Thanks so much, Vanessa, for that lovely introduction. It's a delight to be here to speak at this North-South Ireland meeting, and I think it's an incredible initiative, and to see some of the colleagues from the North as well in the room. So thank you so much to DRNI for organizing. So I'm delighted to share with you today some of my group's work on the effect of risk and protective factors for brain health in midlife. So as we all know, dementia is a global epidemic with huge proportions and effects for individuals, families, and societies worldwide. And closer to home, Ireland by 2050 is projected to have the largest increase in the EU of the elderly population, with age-related costs rising to 16 billion per annum, not accounting for any dementia-related specifically, but even just to consider the costs of the aging population. So it is well accepted now, as compared to even 10 years ago before we started the Prevent Dementia program, that dementia really does start decades before the clinical diagnosis, the onset of clinical symptoms, and that there is a key accumulation of risk burden in midlife. So, and it's very, very important. This understanding has led to an impetus for trying to develop early intervention strategies that can be implemented well before the substantial brain damage occurs by the time that clinical symptoms become manifest. And so what we don't understand from a kind of basic neuroscience perspective, and that's kind of some of the area that I covered that I'll be discussing with you today, is though, however, how do risks impact cognition and the brain in midlife? What are the precise manifestations of risk? What are the mechanisms through which they manifest, so that we can intervene early? And then we understand there are a number of protective factors as well that boost cognitive reserve, but do these protective factors actually start to counteract the effects of risk, again, very early, well before symptoms become evident, and for whom? So these are two questions that I'll touch upon very briefly today, covering some of the work that we've done to understand the impact of risk factors on cognition and the brain, and also to understand whether protective factors can counteract the effects of the risks in midlife, and for whom. And this goes back to a question that was asked earlier: how do I give somebody hope? Well, if we have evidence that actually we can see the evidence on the brain and cognition of protective factors well before dementia develops, and that is one piece of the puzzle in terms of motivating people to take action. And this work is carried out as part of the Prevent Dementia program, which is an international consortium and multi-site study, including 4 study sites in the UK, Trinity College Dublin, and also the INSERM Institute in France. And this is a longitudinal study, so we have been following up a group of 700 individuals who were recruited over 8 years ago, are in midlife, between the ages of 40 and 59 years of age, who at recruitment do not have any cognitive impairment. They are cognitively healthy, and we have now been following these individuals. We are at the 3rd visit at 8 years post-baseline. And we are looking at understanding the impact of risk factors, and specifically who are being most impacted. Where should we intervene? The methods that we're looking at are physical health assessments, lifestyle and cognitive assessments, sample collection, brain scanning, etc. So it's a deeply phenotype study that has the same repeat assessments in the 700 group every single time when we have another wave. So it's a very rich data set that's created over time. This cohort is enriched for family history, or mom and dad. 50% of our participants has dementia, and about 40% of individuals have a lipoprotein E4 carriership, and over 61% are female. In my group, we look particularly, as I mentioned, at the interplay of risk and protective factors, and we look at it from a more kind of basic neuroscience perspective to establish the evidence for some of the epidemiological and policy implications then that we can draw, and that some of which we've heard about earlier today. We'll look at 3 risk factors: the ApoE4 carriership, family history of dementia, and the cardiovascular incidence of aging and dementia composite risk score. We'll look at brain scans, brain imaging, cognition, and a number of what we call protective factors. And in particular with protective factors, in this work, we're hugely interested in those that can be modified in midlife. So we're interested in those specific ones. So I'll tell you a little bit about what I mean by that. We know that there's a number of activities that can protect the brain across the lifespan, and these are known to contribute to what we know as cognitive reserve and resilience, so the ability of the brain to withstand cognitive decline and to withstand the effect of pathology as we age or as we succumb to disease. And importantly, they can play out throughout the life. So education is a hugely important cognitive reserve factor that has lifelong protective effects. The majority of education is accrued in young adulthood and early childhood, and therefore, while education can still be modified in midlife, is not as directly modifiable as some of the other factors that are less understood as well, such as occupational attainment, also another established factor of cognitive reserve, and this denotes kind of complexity of the occupation as well as managerial responsibilities, and stimulating activities, social, physical, and intellectual activities that are also known to have a protective effect on the brain. Most of this work has been done in patients with dementia or MCI, so in clinical populations, and what we wanted to do in the context of brain health is to bring these investigations in midlife before people have any dementia and when people are cognitively healthy, and see how their effects are playing out. And crucially here, we're interested in occupational attainment and the stimulating activities that I will talk about, because people can directly and actively change engagement with these factors in midlife. They can make a huge impact to how much of this cognitive reserve they're able to accrue through these 2 factors. So first, I'll tell you a little bit about a study that we did to find evidence that risk, both genetic, ApoE carriership, and cardiovascular-based risk, does actually indeed directly impact the brain in midlife. And here we took inspiration from neuropathological studies and from understanding built on thousands of neuropathology tracing in the brain that the tau is a key etiological factor in the genesis of Alzheimer's disease. And we know that the tau deposition starts in a small brainstem's nucleus called the locus coeruleus, and then in very early stages of the disease, it progresses, it spreads to the transentorhinal and the entorhinal cortex and the medial temporal lobes. This is work that we did with my PhD student, Fong Dong, and we're interested particularly in this early stage of disease before tau deposition extends to temporal regions and also across the brain. We wanted to find out basically how does the risk for Alzheimer's disease, whether the risk of that Alzheimer's disease is being reflected in changes in the connectivity of this very small locus coeruleus nucleus, which is also a very important neuroadrenaline center of the brain, and the medial temporal lobes, which are important centers for memory. And indeed, we found that when we looked at ApoE carriers, who are represented here in pink, at baseline and the first follow-up wave relative to non-carriers who are in blue, we find that they have a higher connectivity between the locus coeruleus and the entorhinal cortex. So carriers have higher connectivity, which is not what we expected, but then this is followed up with a sharp decrease of the connectivity in carriers longitudinally. And so this is one piece of evidence that suggests that genetic risk for late-life Alzheimer's disease is starting to impact healthy individuals even in their 40s and changing the brain connectivity in centers that are implicated in the genesis of Alzheimer's disease. We also saw that the connectivity between the locus coeruleus and the hippocampus was significantly negatively associated with cognition in individuals who had high cardiovascular risk scores. So the higher the risk score, the stronger the association between the connectivity of these brain centers and cognition. So again, this provides some flavor of, in a very quick summary of what we are finding to be the effect of risk early in midlife on the brain. Another form in which we see that the risk for late life Alzheimer's disease is impacting the brain is in this study that we published recently, again with my PhD student Feng Dong, where we looked at different brain networks shown here in different colors. And here we looked at a property of the brain called functional segregation. And what this denotes is that different parts of the brain, which are known as networks, cohere together. They are, in the healthy brain, they exhibit very tightly interconnected activity. And that is denoted by the colorings here. So we have the motor network in blue, the visual network in darker blue. We have frontoparietal network in red, and so on. And the distinctiveness of this network, so the distinctive clustering of these activities, is hugely important for carrying out different functional processes that our amazing brains perform for us. And we know from large-scale studies is that this segregation of the networks really decreases with age and also is tightly interrelated to dementia severity in dementia patients. So we wanted to know whether the risk of Alzheimer's disease, the genetic risk, is already starting to kind of eat away at this important feature of the brain, which is called network segregation. And again, what we saw when we looked at across the brain is that the carriers in pink have a higher extent of segregation, so a better expression of this feature cross-sectionally relative to non-carriers, but that you can see that if you look at these two time points, that the carriers actually decrease in their segregation over time, but non-carriers don't. And this is more expressed in a particular network that's called the default mode network, which we know from work with patient groups, that is a site of preferential deposition of amyloid beta pathology in the brain, particularly in early MCI and Alzheimer's disease. And what we see again, when you look specifically at the default mode network, is that that carriers have higher segregation of this network, higher distinctiveness of this network at baseline, but then over follow-up, there is a rapid loss of this segregation. This network becomes worse than the feature longitudinally. But this does not happen to non-carriers. So again, this is evidence that the genetic risk is starting to have an early impact in the brain, but also what we are seeing in other domains as well, dichotomous effect of the genetic risk of ApoE in the brain and cognition, which is that a better expression in earlier age groups, which is accompanied with deleterious effects in older groups and also longitudinally. And this is known to relate to the antagonistic pleiotropy hypothesis of aging, and thought to relate to the fact that certain genes have survived through evolution because they confer an advantage in younger ages that then becomes deleterious in older age. But to touch then on the issue of, when we see, again in 15 minutes, I'm going to touch very briefly on the studies that we've published, but we do see that there is an effect on the brain. And what we want to know is whether modifiable factors that are modifiable in midlife can already start to dampen this risk and can provide a buffer against this risk factor. So while the risk is there, can we actually start to hold it at bay, in other words? So this is work that we did with my student Hannigan, and here we're looking at a cluster of activities that are avocational activities that are called stimulating activities, and this is through internationally validated instruments that measure cognitive reserve. And here we are able to capture 7 activities, including exercise, physical exercise, playing a musical instrument, reading, traveling, socializing, engaging in an artistic pastime, and speaking a foreign language. And while some of the effects of these factors, again, are established in patients or in older adults, very little was known before our work on their effect in healthy midlife individuals, like many of us in this room that are going about their lives without any worry about dementia. So we're mostly talking about brain health and kind of looking forward into 3 decades later. So what we did find here is that this higher engagement in this physical, social, and stimulating engaging activities are strongly associated with better cognition. So this suggests that they do actually start to protect cognition already in midlife, even when we don't think we do have a problem. And that what we already also see is that if we look specifically at individuals at risk, such as individuals with a family history of dementia, we see that this effect is even stronger for individuals who have family history. So in the red line here, we see relative to the people without family history is that the people who seem to benefit the most from engagement in these activities are individuals who have mom or dad with dementia. And this is really important, I think, in terms of having implications that we have discussed in published work for encouraging individuals to continue to engage in stimulating activities despite the fear of dementia. A number of people who have parents with dementia are very fearful, become very fearful of dementia, and this has huge psychological implications and often results in people withdrawing from stimulating activities, from social activities, due to fear of dementia. So the message that actually just getting out there and continuing to stay engaged, no matter how comfortable that might be, it is good for you and will protect you, stands a higher chance of protecting you from dementia. I think it's really important that's coming out from this work. And in fact, when we put all of the activities, we put together a group of 17 factors in the context of all the different things that are going on in our lives, including modifiable risks and non-modifiable risks such as sex, age, ApoE. And when we put all of these things together to see actually, what's the overall picture, we see that stimulating activities are the topmost contributor to cognition in midlife, even more strongly so than education, which we know it has a huge impact in lifelong health outcomes. And moreover, it has a bigger impact than the genetic risk for late life Alzheimer's disease. Again, suggesting that engaging with stimulating activities in midlife is a really a great, cost-effective, and scalable strategy for early preventative interventions. And I want to touch very briefly on sex differences that we are seeing in the effect of protective factors. We know that 2/3 of dementia patients are women, and there's a number of unexplored factors that can lead to this huge disparity in the incidence of dementia in women versus men. And there's lots of work going on in this area, but we do find some interesting hints as to what might be part of this picture in our work. So this is work carried out with my student Ching Chi, where we looked at the other element of cognitive reserve, which is occupational attainment, that also is modifiable in midlife. And we're looking at these, as I also mentioned for stimulating activities previously, what is their contribution independently of education. So we factored already education out, and we're looking at what these are conferring outside of that. So when we look at occupational attainment, we don't see an effect for all of the cohort, when we put everybody together. But when we split individuals in sex groups and in carriership groups, we see that females who are ApoE carriers are the only group that show a significant benefit from occupational attainment in relationship to their cognition. So if for females that are ApoE carriers, it seems that occupational attainment is helping to protect their cognition from midlife. And this is the only group, and we don't see this effect in ApoE non-carriers or males who are ApoE carriers. And another kind of piece of understanding in this sex differences comes when we look at actually the deprivation factors, socioeconomic deprivation factors. We heard a little bit about the effect of deprivation. It's a huge area, but again, just to give you a little flavor, deprivations factors are things that actually create barriers to protective factors. And why we're looking at this is particularly because we know that there historically females have incurred a number of barriers to protective factors, including educational historic barriers, which are now equalizing in the Western world, but not worldwide, and are still playing out in kind of older cohorts or aging cohorts. So it's important to still keep that in mind. Women traditionally have the higher caregiver burden across the board, and this leads to less time for engagement with avocational activities of various forms. They have a higher, significantly lower percentage in full-time employment in Ireland today and traditionally, and therefore they have lower occupational attainment overall. So then when we look at the effects of various domains of deprivation on cognition in women, represented here in orange, we have 4 domains of deprivation: deprivation in education, skills, and income, deprivation in occupation, deprivation in income and assets, and then barriers to housing and services, including healthcare. And this is the only one where we see similar levels of effect of deprivation on cognition. So both women and men are negatively affected in their ability to think, in their cognition from this, from lack of access to healthcare and housing. But women are the only group, and it's a huge effect of the negative aspect of deprivation on cognition, and whereas men do not seem to have a relationship between deprivation in education, skills, and income, or training, occupational attainment, and income on their cognition. And these 3 also map very well to the protective factors that I mentioned. So this graph really makes the point that women generally still do not have access to those types of protective factors that we find are so important for protecting brain health. And that might be part of the puzzle as to why we are seeing women continuing to have such a higher incidence of dementia relative to men. So in summary, very, very briefly, I've presented some studies that show that the genetic risk factor and cardiovascular-based risk already starts to have very real impacts on the brain and cognition in individuals who are otherwise cognitively healthy. ApoE carriers show longitudinal declines even within the first wave, which was a 2-year offset from baseline. And ApoE behaves in a dichotomous way, which we are starting to see in other domains of the literature. But what's important is that we are seeing a very real effect of protective lifestyle factors that are modifiable in midlife on the brain and cognition already in this age group. Contributors are educational, vocational, and avocational. I didn't mention education. Education also has an effect, but importantly, avocational activities and vocational activity has a huge impact, independent or with education controlled for. Simulated activities are the topmost contributors to cognition in this age group, and more frequent engagement in simulating activities is particularly beneficial, our data suggests, for individuals with history of dementia, and that higher occupational attainment is associated with better cognition in females, especially in ApoE4 carriers. And women are disproportionately impacted by deprivation in their cognition, so the cognition of women seems to suffer more from deprivation, and we think that's to do with the fact that they traditionally have lacked the opportunity for full engagement in these reserve contributors. So I want to thank my students and staff, my collaborators in the PREVENT family, and all the lovely PREVENT participants, as well as the funding bodies, and thank you for having me today and for listening.